Hi, everyone. Welcome to ID Radio, your go-to podcast for the latest research and essential papers on infectious diseases.
Welcome back to the deep dive.
Mm.
Our job today is to take the absolute mountain of data presented at IDWeek 2025 and, well, try to distill it down to the most influential, practice-changing gold.
It was a mountain, for sure. An extraordinary year for clinical research. We
Right.
saw some incredible randomized clinical trials, the gold standard, that are really redefining how we manage things.
And it wasn't just management. We also saw some critical new antibiotic approvals.
Absolutely necessary approvals, especially in our ongoing fight against antimicrobial resistance.
Okay, so let's unpack this.
Mm.
Because for all this amazing progress on, the treatment and tech fronts, there's a really sobering counterpoint.
Mm.
The staggering resurgence of measles in 2025, I mean, we're talking over 1,500 cases, 3 deaths
Yeah.
in the U.S.
It's the stark, painful reminder that nobody in public health wanted. It shows that all the progress we make with a sophisticated new drug can be, well, completely negated if we fail to uphold basic public health principles.
Like vaccination.
Exactly. It's a constant tug of war.
So that's really our mission today. We want to unpack both sides of that struggle. We're going to focus on 3 big areas. New tools like antibiotics and tech.
Better treatment strategies driven by these rigorous trials.
And then finally, facing those huge prevention challenges head on. So let's start with the tools. The new antibiotics expanding our arsenal.
Yeah, this is exciting. We got 3 new FDA-approved antibiotics this past year, and each one targets a really specific, almost desperate clinical need.
Let's start with the one getting a lot of buzz, Avibactam.
Right, so this is a combination drug, a monobactam paired with a beta-lactamase inhibitor.
And its main approval is for complicated intra-abdominal infections.
Among other things, yes. But the design target is what's so key here. It's built to go after metallo-beta-lactamase-producing organisms, MBLs.
Okay, so for those of us not in the lab every day, can you just remind us why MBLs are such a huge problem?
Of course. I mean, MBLs are these enzymes that bacteria make, and they can just chew up our most powerful antibiotics.
Including carbapenems.
Including carbapenems, which are often our last resort. So having a drug that's specifically designed to bypass those defenses is a major, major victory.
And the trials back this up.
They did, through 2 key studies. The big one, Revisit, showed its overall response was comparable to meropenem, around 68% efficacy.
But it didn't have a ton of MBL cases in it, did it?
That's the catch. It didn't. But that's where the smaller Assemble trial came in. It focused on just 15 patients who were confirmed to have MBL infections,
Right.
and it found favorable responses. So you put them together, and you get this picture that, yes, this is a crucial new option for these highly resistant bugs.
Okay, so number 2 on the list is sulopenem. This one's interesting. First approved oral penem.
It is, and it had a really tricky path to approval.
It failed some of its trials, right?
Yeah, it did. It failed non-inferiority for complicated infections. But in the end, it did prove it was not inferior to amoxicillin clavulanate for uncomplicated UTIs.
So it's got a narrow indication now, but everyone sees the bigger picture. An oral carbapenem class drug.
Yeah.
That's a huge niche for a resistant outpatient infection.
Exactly. It gives us a critical oral step-down therapy we just didn't have. And the third new player, gepotidacin, kind of falls into a similar category.
This one has a totally new mechanism of action, doesn't it?
It does. It inhibits DNA replication in a new way, which is great for avoiding cross-resistance. It also got approved for uncomplicated UTIs.
But the EGL1 trial gave us a big surprise.
A huge surprise. It was non-inferior to the standard of care injectable ceftriaxone plus oral azithromycin for uncomplicated urogenital gonorrhea.
That is massive. I mean, with gonorrhea resistance climbing, getting away from injectable ceftriaxone for everyone would be a game changer for clinics.
No question. So the takeaway for these last 2 is that while their current indications are uncomplicated, their real strategic value is future-proofing our ability to treat resistant bugs with oral options.
Okay, so let's pivot from new tools to new approaches.
Right.
Let's talk about the DOTS trial using delbavancin for S. aureus bacteremia.
Right, so this is all about long-acting antibiotics. The trial looked for an alternative to the usual 4 to 8 weeks of 5e antibiotics these patients need.
A huge burden on patients. BICC lines, daily visits.
A massive burden. So the intervention was just 2 doses of IV delbavancin given 1 week apart.
And the difference for the patient is, well, it's night and day. What did it show?
It didn't prove superiority, so it wasn't better than standard therapy, but it did hit that crucial goal of non-inferiority for clinical efficacy at day 70.
So just as good, but infinitely easier.
That's the key. Now, we have to mention the caveat. This is only for carefully selected patients. They excluded people with endocarditis or infected hardware they couldn't remove. But for the right patient, this is a massive logistical shift. It could let them avoid a long hospitalization.
Okay, next up, the microbiome, fecal microbiota transplantation, or FMT. We know it's good for recurrent C. diff, but what about as a primary first-line treatment?
Yeah, this was a fantastic RCT, a head-to-head comparison. One-time FMT enema versus 10 days of oral vancomycin for a first episode of CDI.
And they actually stopped the study early.
They did, because the data clearly showed non-inferiority. The initial cure rate was similar, 67% versus 61%.
But that's not the whole story, is it? It was the recurrence rate that was so compelling.
Exactly. The difference was almost entirely driven by the dramatically lower recurrence in the FMT group. We're talking 5.6% versus 21% in the vancomycin group.
Why?
A primary treatment that cuts recurrence by nearly 3/4. That is transformative.
It really is.
Yeah.
Okay, one more in this section. The step-up RCT for bacterial vaginosis, or BV. This was a surprise because treating male partners has failed before.
It has. But this trial changed the strategy. They treated male partners with 7 days of both oral metronidazole and a topical clindamycin cream.
And the result?
Dramatic. Partner treatment led to a significantly lower BV recurrence at 12 weeks, 35% recurrence versus 63%. A hazard ratio of 0.37 is huge.
That's a phenomenal clinical result. But let's be realistic. Getting a partner to adhere to 2 different drugs sounds difficult.
It does, and the researchers acknowledge that. This is a classic case where we've proven efficacy in a trial, but figuring out the real-world public health delivery, that's the next challenge.
And that leads us perfectly into our next section, antimicrobial stewardship. The whole philosophy here is shorter is the new long.
It really is, and in 2025, we finally got the huge data sets we needed to challenge some of these long-standing traditions.
Starting with the Balance RCT, over 3,600 patients. This was the big one for bacteremia duration.
It was. It asked a very simple, crucial question. For non-S. aureus bacteremia, is 7 days of antibiotics as good as 14?
And the answer was?
A resounding yes. 7 days was non-inferior to 14 for 90-day mortality. The rates were basically the same, 14.5% versus 16.1%.
That's a massive practice change. It supports a major shift away from just tradition.
It really does. It's a huge win for stewardship.
So moving from general guidelines to personalized care, let's talk about the ADPT sepsis RCT, which used biomarkers in the ICU.
Right. This trial compared standard fixed-duration care to using either procalcitonin or C-reactive protein CRP to guide when to stop antibiotics.
And the results really highlighted how specific these tools need to be. What worked?
The procalcitonin-guided group, using PCT, significantly reduced the total antibiotic duration, and it was proven non-inferior for 28-day mortality.
So it's an effective, personalized tool.
It is, but the CRP group, which is a much more general marker of inflammation,
It failed.
It failed, it didn't reduce duration, and they couldn't prove it was non-inferior for mortality. So it tells you, you can't just use any inflammatory marker, it has to be the right one.
Okay, finally, let's talk about the dilemma from the kids' febrile UTI trial, the NDI-UTI RCT.
Right, so this compared a fixed 10-day course versus an individualized approach where you stop 3 days after symptoms resolve.
And the individualized approach had some real benefits.
It did. Significantly fewer antibiotic days 6 versus 10 and fewer side effects like rash or diarrhea.
But there's a big but here, isn't there?
There is. The personalized approach failed to meet the non-inferiority margin for UTI recurrence.
What were the numbers?
Recurrence was 11% in the short group versus 6% in the 10-day group. So you have to weigh fewer side effects against a 5% higher chance of the child getting sick again.
And that's a tough decision for clinicians and parents. It makes widespread adoption really difficult right now.
It really does. It shows you how messy this can be.
So where does technology step in to help us bridge this gap? Let's talk technology and innovation.
AI is really starting to help us practice smarter stewardship right at the point of care.
Like with the Inspire 3 and 4 trials, they used AI within the electronic ordering system.
Exactly. These were cluster randomized trials. An AI model looked at over 60 patient variables to predict the risk of a multi-drug resistant organism, an MDRO.
And if the patient's risk was below 10%.
The system flagged them as low risk, and when the provider went to order antibiotics, a prompt would pop up recommending standard, narrow-spectrum drugs.
And it worked.
It worked great. Significant reductions in broad-spectrum use, 27% for skin infections, 35% for abdominal, and crucially, with no increase in length of stay or ICU transfers.
So it helps you treat smarter without compromising safety.
Perfect.
Now let's look at tech improving access for the most vulnerable. Tell us about the Peer Tele-HCV RCT.
This focused on a really hard-to-reach population, people who use drugs experiencing houselessness in rural Oregon.
The barriers to care there are just enormous.
They are. The study compared standard referral to a peer-assisted telemedicine model for hepatitis C treatment, and the impact was staggering.
How staggering.
Treatment initiation jumped from just 12% in the standard care group to 85% in the telemedicine group.
Wow.
And that translated directly to cures. 63% viral clearance versus only 16% in the control group. It's just a profound example of combining tech with trusted peer support.
That model could change how we tackle epidemics in vulnerable populations everywhere. Okay, finally, the NUG-FLU chronic RCT in Denmark.
This was a huge pragmatic study. Nearly 300,000 high-risk citizens. They tested different electronic letters based on behavioral science to try and increase flu vaccination.
And all the nudge letters worked better than usual care.
They did. The highest rate they achieved was about 42% vaccinated, compared to about 28% in the usual care group. It's a cheap, easy intervention.
But even with a smart, affordable intervention targeting a high-risk group, we're still well below 50% uptake.
Exactly. And that sets the stage perfectly for our final section, prevention challenges.
Let's start with a success story, though. The Ebola vaccine during the DRC outbreak. They used a ring vaccination strategy.
Right. Instead of mass vaccination, you vaccinate the contacts of confirmed cases. And the data from over 300,000 vaccinations confirmed it works.
How well?
Contacts vaccinated within 8 days of an index case had a significantly reduced rate of developing Ebola. It really proves the concept is feasible even in very challenging settings.
Okay, so that's the good news. Now we have to circle back to the bad. The measles resurgence. We are seriously flirting with losing our elimination status in the U.S.
And it's clearly driven by falling MMR vaccination rates, which are now below that 95% herd immunity threshold.
So what does the future look like? There was that fascinating modeling study by King and company.
Right. They look 25 years ahead. And because measles is so incredibly contagious, they predicted that just a 10% further drop in vaccination rates would be enough to make measles endemic again within a decade.
Endemic, meaning it's just always circulating. A terrifying thought.
It is. But the model also gave us a target. It showed that an increase of just 5% in vaccination rates could prevent that outcome.
So it gives us a clear, achievable goal for public health intervention.
Exactly. A window of opportunity.
Okay, let's turn to another huge global threat. MDR tuberculosis prevention. We had 2 big trials, VQUI and TB Champ.
Right. Both looked at using levofloxacin to prevent TB in household contacts of known MDR TB patients.
But individually, the studies were underpowered.
They were. But a pre-planned meta-analysis of the combined data found a 60% relative reduction in TB incidence. That is a highly meaningful level of protection.
Is there a safety trade-off we need to know about?
There is. The meta-analysis flagged an increased risk of musculoskeletal events, especially in people aged 10 and older. So it's a powerful tool, but you have to use it carefully.
And we should probably mention the cultural impact here. Advocates like John Green bringing so much attention to TB. It highlights the need for continued public support.
It really does. And I think when you look across everything from 2025, the theme of progress is just undeniable. The ID field is constantly improving care, whether it's new drugs, smarter stewardship, or new care delivery.
What really stands out to me, though, is the contrast. The speed of these advancements versus our vulnerability to just fundamental public health setbacks, like measles. I mean, we've proven that a sophisticated algorithm can predict MDRO risk in a hospital, that telemedicine can cure hepatitis C in our most vulnerable populations.
Which brings us to the critical question we have to leave with.
Exactly.
Given these incredible technological leaps, why does the simple behavioral hurdle of encouraging an affordable, life-saving flu shot still leave us below 50% uptake in a high-risk group? Closing that gap between our clinical tech and our public health adoption, That remains the single most important challenge for the future.
A profound challenge to consider as we close this deep dive.
Thanks for watching. Please make sure to like, share, and subscribe to ID Radio.